Good morning. Thank you for the introduction, and thank you to the organizing committee for giving this opportunity to speak about my research. And like he said, I'm going to present my dissertation research on American botanicals as possible alternatives to hormone therapy during menopause. So I'm looking at American Indian medical ethnobotany because 2,800 plants from American Indians have been specifically used for medicinal purposes. And in the industry today, 5 of the 10 highest selling herbal supplements have been traditionally used by American Indians. And also, American Indian ethnobotany has contributed to some pharmaceutical advantages, such as this compound here, also known as etoposide phosphate, which has been used to create derivatives from it for anti-cancer drugs. And this was from looking at American Indian ethnobotany from the plant Podrophyllum peltatum, also known as mayapple. So the big M, menopause, right? Everybody knows somebody that's experienced menopause or experiencing menopause, and here it's shown in a comedic way. But for some people, it can be detrimental to life or it can affect the way that they live. And so menopause is inevitable for all women who live past middle age. And basically during menopause, it is when women experience low levels of estrogen, and as a result, they may have symptoms such as hot flashes, night sweats, they can't sleep, they have vaginal atrophy, and even mood swings. And so a lot of women try to help these symptoms by taking drugs or other things for that. And so one of the drugs that women would take is hormone therapy. And hormone therapy is considered to be exogenous estrogen or estrogen plus progestin. And here, in 2002, hormone therapy hit the Time magazine. And it states here that the truth about hormone therapy, hormone replacement therapy is riskier than what people think, what women should do. And what a lot of women did was that they resorted to botanical dietary supplements because the World Health Initiative came out with their results from their study in 2002 stating that hormone therapy caused increased risk of coronary heart disease, breast cancer, and even stroke. And some of the botanicals that women would take are, black cohosh, wild yam, and red clover. And these 3 botanicals have been specifically traditionally used by American Indians. Now, there's a whole slew of traditional plants for women's health by American Indians that are under investigated. And so the purpose of this project is to to figure out more if these other botanicals can have efficacy and also are safe for women to use outside of the most popular ones. So phytoestrogens, these are the compounds that are present that are considered to be the active components that women would use for menopause. And so phytoestrogens are compounds that mimic the effect of endogenous estrogen. So it can have estrogenic activity in some tissues and block the effects of estrogen and have anti-estrogenic activity in other tissues. And so this tissue selectivity allows phytoestrogens to be more beneficial for women's health compared to hormone therapy. And so, for example, anti-estrogenic phytoestrogen in breast tissue reduce the risk of hormone-dependent cancers, where estrogenic phytoestrogens can aid with menopausal symptoms and help maintain healthy bone density. So the purpose of this research was to support American Indian use, understand botanical therapies for menopause, identify natural products for cancer prevention as well as inflammation, because we see that estrogen can have an effect on these 2 things. And also my hypothesis is that some botanicals historically used by American Indians will demonstrate estrogenic cancer prevention or anti-inflammatory activities that promotes women's health while also protecting women from estrogen carcinogenesis. So this project was done in collaboration with the Chicago Botanic Garden. They gave us over 160 plant species, and my advisor, Dr. Soejarto, he selected 15 species to look through based upon their taxonomy, their availability, and also their chemical properties. And so these are the 15 species that he selected. And what I did is went through this whole process of, first of all, figuring out what these species are by doing a lit review, what activity they have, and also what traditional uses are there. And then I narrowed it down to 3 species to start the initial examination. And then I did the biological assays, and from that I selected the active species, and then did bioassay-guided fractionation on the active species to figure out what the active compounds were. And now I'm working on figuring out what these active compounds are. So to begin, I selected Amorphokinesis, also known as lead plant. That's here. We have Cyphium perfoliatum, also known as cup plant, and Echinocytus lobata, also known as wild cucumber. And these how the samplers look when I receive them. They're grounded up and they're in fluorescent tissue. And I use two databases to help me figure out what the literature stated about them and also what were the traditional uses. And so the first database that I use is called NAPAlert, or Natural Products Alert, and that's housed at UIC. And it provides pharmacological data, biological activity data. It also gives traditional uses of the plants and also the chemistry. And I also use Dr. Dane Morgan's database, maybe Ethnobotany, to give me the traditional uses of these plants. And so from that, I was able to see that lead plant has some traditional uses as an anti-rheumatic, so I'm thinking about that it may have anti-inflammatory activity. And it also has some carcinogenesis inhibition. Cup plant didn't have any biological data out there. However, it has several traditional uses for women's health, as seen here. And wild cucumber has been used as an abortifacient, but also has some biological activity for anti-inflammatory activity. And so that's why I chose these plants. The second step was to do a ethnobotanical review, and this is what I'm currently working on today. So I am reviewing some literature, but also I'm working with the American Indian women population in Chicago. So first to see if these three species and other species that I'm working with have been traditionally used for women's health and menopause, and to identify other botanicals for women's health and menopause. And also, when I did a class last semester, I found out that American Indian women perception of menopause is not mentioned in literature. So I decided to add that to my interviews. I'm also working on a review paper, and it will be submitted into the phytochemistry reviews on American Indian botanicals for women's health, and so please look out for that at the end of this year. Second, I did the preliminary evaluations looking at estrogenic activity and cancer prevention activity. And I did a methanol extract on the inflorescent tissues as seen here. And I did the Ishikawa assay to identify potential uses for menopause, EQ1 induction assay for cancer prevention, and the KEEP1 modification assay also for cancer prevention. So the results from the Ishikawa assay, we see that lead plant has strong anti-estrogenic activity. However, we see that it's slightly toxic. And we also see that cup plant has some anti-estrogenic activity, and wild cucumber didn't have any significant activity at all. We're going to get back to this. And for the cancer prevention potential assays, in the NQ1 induction assay, we see that lead plant have some dose-dependent chemopreventive activity, but it's slightly low, and also is slightly toxic towards the cells. And in the KEEP1 modification assay, so here we're looking at how the KEEP1 protein is being modified. And as you can see here, you see a difference in the mass of the KEEP1 protein in comparison to the control, the actual KEEP1 protein. And here, none of the three plant species had an effect on the KEEP1 protein, resulting in no chemopreventive activity via this KEEP1 NRF2 pathway. So to sum that up, I basically showed that lead plant has some anti-estrogenic activity, but it's slightly toxic. It also has some cancer prevention activity, but it's slightly toxic to the cells, and so that may skew the results. While cucumber didn't have any activity in these three assays, and the cup plant extract had some anti-estrogenic activity. So I took a chance and I decided to stick with lead plant. And the reason I did that is that I was hoping to separate out the toxic components from the active components. And so I did liquid-liquid partitioning, basically separating out the compounds from polar to non-polar with water and acetate, chloroform, and hexane. And what we were able to see from the active partitions, so this is our methanol extract here, we were able to see that the chloroform partition was able to retain dose-dependent anti-estrogenic activity. The hexane did not have any dose-dependent anti-estrogenic activity, but it was still active. And this was much higher than our methanol extract. I was also able to do the same assay, look at it from a different way. So I looked at it for its estrogenic components in the presence of estradiol, which is a positive control for estrogenic activity. And so estradiol, we see here, has 100% activity, but in the presence of our extract, it's pretty much staying the same. We want that to go down. So for our chloroform partition, we see that estradiol, estrogenic activity has been diminished with the chloroform partition and also with the hexane partition. But when I later did the cytotoxicity test, we see that the hexane, which I'm not showing the data, but the hexane partition was the one that had the toxic components, whereas the chloroform partition was non-toxic. So I decided to stick with this chloroform partition for my bioassay-guided fractionation. I also went ahead and examined the extracts in the partition in the anti-inflammatory assay known as the Griess assay, and we see that there's some anti-inflammatory activity in the methanol extract, some dose-dependent anti-inflammatory activity in the hexane partition, and some in the left chloroform partition. So now I'm working on bioassay-guided fractionation, and this is the process that I'm at now, trying to figure out the active components using the Ishikawa cell-based assay. So after each step with fractionating, I go back and do this assay to see where the active constituents are. I'm also working on an assay to give me a quicker identification of the antagonist ligands by using mass spectrometry, and I'm hoping to use the active compounds into these assays here to figure out and make sure that their active constituents are active. So, thank you, and I would like to give thanks to everybody that helped me.
Okay, we can do questions.
The growing of plants at the Botanic Garden is somewhat artificial, and I'm wondering if some of the traditional ecological knowledge or larger growing areas, natural, would have an effect on different actives that might be present, and I wonder if you've looked at that.
So that's with any botanical that anyone works with when doing laboratory assays. There always has to be some type of chemical fingerprinting and authentication and making sure that what we're looking at is actually from the actual plant. And I'm working on doing that with our center. I worked on DNA barcoding, which I haven't shown here, to make sure that the plant is actually what it is, and also doing the chemical profile and sampling different plants from different areas. I was able to grow plants in our field station in our active garden. So those are 3 different samples that I can collect data from to compare and contrast the chemical constituents to make sure that this is exactly from this plant. But whenever you're working with botanicals, you always have to verify that it is what it is. And you may see those differences, but there's a baseline to that when you're working with botanicals.
Can we go on more?
Yeah, we can do.
Sonia Peter from Barbados. We know that in traditional usage, or at least we think so, that in using an extract from the plant as is, there could be synergistic action of the natural agents. Are you hoping to look at an isolate, or maybe look at the activity of the fraction as it is?
Both. If I can have retained activity when looking at an isolate, then that's perfectly fine. But if, and let's say, like one of the fractions is the best, has the best activity, and if I can describe the compounds in that fraction, that's good enough for me. Because sometimes it does not always work with one active compound. And so I thought I'd put compounds on there just in case.